Hello everyone, welcome to our next video. Today we are going to have the second part of the types of amino acids, and the topic of our discussion today will be the uncharged polar amino acids. We discussed about the non-polar amino acids in our last video in the part 1 of types of amino acids. Today is going to be the part 2. So stay tuned and check that out. So, before I begin with the amino acids, let us understand what is polarity. Since we are discussing about the polar amino acids, we must understand what is polarity and why does polarity arise. In the last lecture, we discussed about non-polar. So, what does non-polar mean? We need to discuss about that first. So, polarity arises when there is difference in the electronegativity between two atoms that are bonded together. So, just imagine, for example, we have oxygen bonded to hydrogen. Since oxygen has higher electronegativity than hydrogen, so oxygen will be pulling the electrons towards it. As a result, a dipole moment is created where the density of the charge, the electrons, is more towards one atom than the other. That creates a polarity of charge where two poles are created. One is partially negative and one is partially positive. So these sort of difference in charge or the amount of electrons present on the two sides of two atoms is what causes the polarity. So for this thing to happen, we must have two atoms bonded in which we will have difference in the electronegativity. Okay, so among the amino acids, the uncharged polar amino acids, we have 6 of those. We have 6 uncharged polar amino acids, and let us see how this become polar. They will have no charge, but they will still be polar, means there will be a difference in the density of electrons between atoms. So let's get through those. The first one in the group is serine, S-E-R-I-N-E, serine. And what happens in serine is it looks very simple like all the other amino acids, but in its R group, this is the R group of serine, and in the R group we have one hydroxide group attached. Now, hydroxide group is a polar group. Why? Because oxygen has a higher electronegativity compared to hydrogen and the carbon. So, since there is a polar group attached to this whole compound, the electrons will be more dense towards the oxygen. So, in the oxygen, we will have more density of electrons. Also from the hydrogen, the electron that comes will be attracted towards the oxygen. As a result, oxygen will have a partial negative charge, causing a polarity, and hydrogen will be having a partial positive charge. So this will create a polarity in this region that makes this uncharged amino acid a polar amino acid, because oxygen has higher electronegativity compared to the hydrogen. So I hope that is simple. It is not that tough, just need to understand it in a simple way. We have, okay, let me erase this part. Better understanding, we have oxygen and we have hydrogen bonded. This is bonded to C. Now, since oxygen has higher electronegativity means the desire to gain electrons. Electronegativity is the desire to gain electrons. So since oxygen has more desire to gain electrons, the electrons present in this bond will not be at the center. They will be attracted more towards the oxygen. Similarly, the electrons, the pair of electrons in between C and O will also not be at the center. They will also be attracted towards oxygen. As a result, oxygen will have a partial negative charge, hydrogen will have a partial positive. So since this portion is having a partial negative charge and these are having partial positive charge, there occurs a polarity of charges. Just like North Pole and South Pole. Here also we have this negative pole and the positive pole, opposite poles creating polarity. So that's why this serine is polar. I hope that is clear. Let's move on to the next one, which is threonine. In threonine also we have one hydroxide group attached. Look at this. This is our hydroxide group. Since the hydroxide group is here, the same condition applies, and this will also have polarity. Now, what is the difference between serine and threonine? In serine, we have one hydrogen, and this hydrogen is replaced by a methyl group in case of threonine. Not much of a difference. If you can remember this structure, you will easily be able to remember this threonine structure. Okay, this one contains one hydrogen here, one hydroxide hydrogen and another hydrogen, second hydrogen is attached to this carbon. In this case we have hydroxide hydrogen and one methyl group. Okay, now let's move into the next one, which is cysteine. I hope you remember in my last video I said there are two amino acids only which contain sulfur. One was this cysteine and the other is the methionine. So methionine we studied in our non-polar amino acids. Hope you remember that one. It contains one sulfur. So here also, let me erase this part. This side chain contains one sulfur which is at the terminal end, and since it is at the terminal end it will show polarity. So these are the first three. We have three more, so let's look into those as well. These are our next three amino acids that we are going to talk about in the uncharged polar amino acids. So next one is asparagine. Now asparagine, these two are different from the previous one. Asparagine, this is the R group. Here we have one oxygen bonded to this CH2 and one nitrogen bonded to CH2 and as well as H2. Now oxygen is highly electronegative, so it is going to pull the electrons towards it, plus nitrogen is also electronegative compared to carbon and hydrogen. So nitrogen will also pull the electrons towards it. As a result, this will become partially negative and the nitrogen also becomes partially negative. We have a dense charge of electrons towards oxygen and nitrogen, causing this to be a polar amino acid without any charge. See, these are partial negative charges. This charge is present inside the atom. It is because in the bond between the two atoms, the electrons are shifted towards this. It is not like it has taken all the electrons. The partial power to draw the electrons towards itself is more for nitrogen. So that's why it is a partial negative charge and this is partial positive charge. I hope that is clear. It is not that complex of a thing. Okay, so this one drives the electrons. The same case occurs in glutamine. Now glutamine and asparagine also have somewhat similar structure, only little bit small differences. Here we have only one single CH2 group and oxygen NH2. Here we have CH2, CH2, C. So we have 3 carbons extra after the central carbon. Here we have only 1 extra carbon. So remember 3 extra carbons. Obviously hydrogen will fill up the remaining spaces of carbon's valency. So if you don't have to remember the number of hydrogens, you only remember the number of carbons. Here we have only 1 carbon. Here we have 1, 2, 3 carbon. And the third carbon we are going to have double bond O and NH2, double bond O, NH2. Same case, oxygen is more electronegative, pulls more electrons towards it. Nitrogen is more electronegative, pulls more electrons. As a result, these will turn out to be polar. So that's why this asparagine and glutamine are polar amino acids. Now let us move into the last one, which is the tyrosine. Tyrosine has 2 properties. The first thing is it contains a hydroxyl group similar to the first 2 amino acids that we saw. So like those, we have hydroxide in this, like serine and threonine. Hydroxide group, so oxygen will be drawing more electrons towards it, hence it becomes polar. Very simple. This is our R group, the side chain. So this draws more electrons towards it and hence it becomes more polar. Now there is another special property of tyrosine. Tyrosine is present in whichever amino acids they are in whichever proteins they are present, they absorb light, just like the other two, tryptophan and phenylalanine, because it contains one aromatic chain. Since it contains this aromatic ring, sorry, not chain, ring, since it contains this aromatic ring, it will have the property of absorbing light and giving peaks of light absorbance, which we will study about this later on. Just for now, remember the tryptophan, tyrosine and phenylalanine, these three amino acids help in absorption of light, means not help in, they absorb light, and that helps us in detecting the property of the amino acids, and this is because it contains one aromatic ring. Okay, so these are our 3 and the previous is a total of 6 amino acids which are uncharged but polar. I hope the structures are simple to remember. You don't have to remember all the structures, just remember it's like these two. For example, these two, remember the structure of asparagine, which is the simpler one. We have CH2O NH2, and in order to remember glutamine, remember that asparagine and glutamine are they are close relatives, maybe brothers or sisters, or maybe the parent, the mother and the son, or mother and the daughter, and they are closely related. As a result, we are going to have slight differences, although they are going to look almost the same, just like two brothers who look almost the same but are not really same. So just like that, they are almost similar, just we have 3 carbons attached to this carbon, central carbon. Here we have only 1 carbon. Okay, similarly in serine and threonine also there is similarity in the structure. So if you go towards learning the structure of serine, you can know the structure of threonine. Similarly here asparagine and glutamine are closely related. So all the structures likewise are very closely related, so you don't have to take the pressure of remembering everything. So remember one, you will remember the other automatically. Okay, so here we come to the end of this lecture. This is our second part. We will have a third part as well. In our next video, we are going to study about the charged polar amino acid. Here you have done uncharged polar. We will move into the charged polar amino acids in our next video. So I hope it was useful for you. I hope you understand it clearly. If you don't understand, as usual, put your comments in the comment section so that I can help you answer your questions. If I share it on Facebook or WhatsApp and wherever I will share, you can ask me questions on those platforms as well. On Instagram, you can follow me there, and hope you enjoyed the video. See you in the next one. Peace.